Oh, okay, very good morning, my dear friends. Cpget free online coaching for botany and plant sciences. So today's our eminent resource person, Dr.M.Rambabu garu, Assistant Professor, Department of Botany, one of the prestigious institution in Warangal, that is Kakatiya Government Degree College. So Sir is going to uh, start the session about the plant tissue culture and biotechnology, one of the important area. So my dear friends, listen during the class. If you have any doubts, please ask our eminent resource person, Dr. M. Rambabu garu. Sir, please welcome and start your session and share your screen also.
Professor, thank you for your introduction. Now, please give the share option.
Yes, sir, I have already given to you, sir.
Right. Good morning. Good morning, all. Now, my topic is plant tissue culture. So, this is very, very important and very, very interesting topic in our entire botany. So, I'm expecting one or two questions may come from this plant tissue culture syllabus. I will give the complete information and also notes to you. Don't worry. So, I will screen share. Madams from Telangana Social Welfare Degree College, Dr. Ujjwala Madam and Dr. Deepthi Madam, and my dear students, I will give some important inputs regarding plant tissue culture. Within 5 to 10 minutes, I will take the class. Okay, plant tissue culture is actually the culture of plant material. The culture of plant material. The culture of plant material on suitable nutrient media is called as plant tissue culture. Plant tissue. Either tissues or parts of the plants. Parts means seed, leaf, stem parts, root parts. So whatever it may be, culturing of tissues or parts on suitable nutrient media is called as plant tissue culture. By that process only, we can produce the complete plantlets from the test tube on nutrient media. This entire plant tissue culture process is first carried out by the great scientist Gottlieb Haberland, scientist from Germany. Haberland scientist, he carried out the first the plant tissue culture technique, and he produced the complete plantlet from tissue culture process. So that's why he is called as a father of plant tissue culture. Who is the father of plant tissue culture? Haberland. This is, I think, important question for the purpose of entrance examination. Who is the father of plant tissue culture? This question may become in entrance examination. Culture technique is this is completely in vitro technology. In vitro means inside of the laboratory, in the test tube. There are two words are there, in vitro and in vivo. In vitro means inside of the test tube, that is inside of the laboratory. In vivo or ex vitro word is there, another word opposite of in vitro. That is in vivo, outside of the laboratory or outside of the test tube. So this tissue culture process is a complete in vitro technology. This is inside of the test tube and inside of the laboratory, and always under aseptic condition experiment. This experiment is always under aseptic condition. So one more thing is the tissue culture technology based on cellular totipotency. Cellular totipotency. Completely this tissue culture based on cellular totipotency mechanism. What is the cellular totipotency here is each and every cell, plant cell or animal cell, each and every cell have the capacity of production of new plant. Each and every cell have the capacity of production of new plant, which is similar to its parent plant. So that capacity is coated on its own genetic material. So this capacity is called as cellular totipotency. Cellular totipotency means each and every cell have capacity of production of new plant, which is similar to its parent plant. That capacity is coated on its own genetic material. So by the repeated 3 actions, these plants are going to regenerate into new plantlets. These tissues, these parts are tissue cultured tissues or parts are regenerated by 3 steps: cell division, cell elongation, and cell differentiation. There are 3 steps are there. My dear students, listen carefully. By the 3 steps, these cultured tissues are regenerated into new plantlets. What are the 3 steps, 3 actions? One is cell division, second one is cell elongation, and third one is cell differentiation or deviation. So first step is cell division. By the affecting of the availability of elements, micronutrients or macronutrients, whatever it may be, by the availability of the elements, the cultured cells are further goes to division, cell division. The single cell divided into new daughter cells. That is the first step in the plant tissue culture. So by that only multiple cells will be developed from the inoculated tissue. The divided new daughter cells are next goes to size increasing. That is cell elongation. Now mother cell divided into 2 daughter cells. These 2 daughter cells further goes to cell elongation. So then further step is cell differentiation. That elongated new daughter cells are converted into, take a part into plant parts. Some cells are converted into roots. Some cells are converted into stem parts. Some cells are converted into, leaves. By that only deviation, the complete plantlets will be developed from the callus. So these are the 3 steps: cell division, cell elongation, and cell differentiation. From these 3 steps only, these inoculated tissues are further regenerated into complete plantlets. This is the theory. For the purpose of these 3 steps, 2 basic plant growth regulators plays a valuable role. By the 2 plant growth regulators only, the cell division, cell elongation, cell differentiation will be takes place. What are the 2 important plant growth regulators? Here, this is the question. auxins and cytokinins. Auxins are used for cell elongation. Auxins are used for cell elongation, and cytokinins are used for cell division and also cell differentiation. Now, first step is cell division. For the purpose of cell division, cytokinins are useful. Cell division, cytokinin. Then divided new cells or further goes to cell elongation. That for the purpose of cell elongation, auxins are useful. Then for the purpose of cell differentiation, again cytokinins are useful. This is very, very important. In your entrance examination, these kind of questions will become. For the purpose of cell differentiation, which type of hormone is useful? This is a question. Auxins, cytokinins, gibberellins, and abscisic acid, both options will be given. Cell differentiation means the divided elongated cells are take part into the leaves or stem or roots or internal parts of the plant. That is the deviation. So this is important question. No, the plant tissue culture based on cellular totipotency, then this is possibility by the three steps, three stages. They are cell division, cell elongation, and cell differentiation. So for the purpose of these three, auxins are useful for the cell elongation, and cytokinins are useful for the cell division and cell differentiation. So this is the theorem. Then coming to plant tissue culture process is completely lab to land program. You know, inside of the test tube, the new plantlets will be developed by the plant tissue culture. Inside of the test tube, new plants developed. From the test tube, the new developed plants will be transferred to the land. By that only, our complete experiment carried out. Lab to land. Inside of the test tube, new plants developed by the taking of different types of the techniques. The test tube plants transferred to the land, that is lab to land. The process of lab to land program is called as hardening and acclimatization. This is the question. What is hardening? What is acclimatization? The test tube plant is transferred to the normal environment, normal field, that is lab to land. This process is called as hardening. The test tube plants immediately not transferred to the field. That is a highly risky process. The test tube plants is present inside of the test tube. It has some glass around its environment. Inside of the glass only, test tube only, that plant is surviving. Suddenly, the test tube plant picked up and transferred to the field. It is not possible. Why? Because the test tube plant there is no cuticle layer around the test tube plant. Remind, inside of the test tube plant, there is no cuticle layer around the epidermis. For example, you take the section cutting, that test tube plant, take the section cutting and observe under microscope. There is no cuticle layer around the epidermis. But you see outer side environment plant, you know, take the outer side environment section cutting of the stem, just you observe thick cuticle layer is present around the epidermis. So this is the difference between the tissue cultured plant inside of the in vitro plant and outside ex vitro plant. The cuticle layer is absent in test tube plant, cuticle layer present in outside environment plant. So this test tube plant gradually developed the cuticle layer around the epidermis and gradually shifted to the outer field. The process is called as hardening. తెలుగులో దాన్ని దృఢీకరణ అంటారు. ఇంగ్లీష్ hardening means the plant is converted into hardening nature. It will be take some hardening nature. Come step by step. It will be taken from the test tube, wash the media from the roots, then put into soil, right, and kept into greenhouse. Then gradually, after some days, shifted to the normal environment condition. So the entire process is called as hardening. Here the question is, the test tube plant is converted into means transfer to the field. That process is called as hardening. Or one more word is acclimatization. Acclimatization. This is the question. And this is also called as micropropagation. Tissue culture process is also called as micropropagation. Small sized propagation production. The small sized tissue can produce number of plantlets in the low time period. Here, the small sized tissue can produce high number of the plantlets in low level of time period. So this is called as micropropagation. So this is a small introduction about the plant tissue culture. So I think some of the students feel very tired taking some class. Tomorrow, 9 o'clock, I will take the perfect class. Thank you, my dears. Thank you, sir. Tirupati, sir. Hello, Tirupati, sir. Tomorrow I will take the class, regular class.
Thank you, sir.
Tirupati, sir.
Okay, sir. Thank you. Thank you very much, sir. So tomorrow, 11 o'clock, sharp 11 o'clock, your
Okay.
session starts from your session, sir.
Sure,
Sir, thank you. Thank you very much, sir. Thank you.